Hi guys, I just wanted to give you a brief overview of some of the things for the renal slide. I know Dr. Pearl covered this with you guys, but there's a diagram that we have posted on Blackboard that I think is really useful. So that diagram's here, the scan of notes from the renal lab. And if you click on that and open it up, what you'll find is a document that looks something like this, where if you scroll down, you can see there's multiple different levels to it. Um, the point of this is basically to help illustrate that there's two major compartments that we can think about for the body. The intracellular fluid here is depicted by a single box, and then the extracellular fluid is shown as this box out here. And this is kind of reflecting on probably something that you saw from your slides that you have for from 291. But in case you haven't seen that, let's pull it up real quick. And that's also on Blackboard for you. One moment. There we go. So in the first slide here, they talk about how we have these different fluid volumes in the human body, and that 2/3 of the total volume in our body is in the intracellular fluid, and that 1/3 in the extracellular fluid. So what we did in the lab, or what you guys did in the lab last week, was learning about how changes in the osmolarity or in the blood volume can ultimately cause changes in how the body works and how it fires, or the types of reflexes that can end up occurring. So if we kind of scroll through some of these slides, this talks about the basic nephron reabsorption secretion type of work. This talks about the action of vasopressin here. But I want to zero in on these two slides here, slides 6 and 7, because what this does is it outlines the different reflexes that help us to be able to control and maintain homeostasis for renal function. So the first one's basically talking about the effect of osmolarity and what can happen. And so if we kind of zoom in a little bit here, what we're looking at is, let's say that there's homeostasis here where we have a normal amount of sodium concentration in the ECF, and then there's a disturbance, a decrease in the sodium concentration in the ECF. What then can follow is that will inhibit the osmoreceptors. So that's a key thing to remember, is that an inhibition of the firing of the osmoreceptors causes the next couple of steps to happen. And the things that happen in response to that is when we have our osmolarity drop below 280 milliosmoles, is that we have a drop in the antidiuretic hormone, and another name for ADH is vasopressin. And then we also have a drop in thirst. So those are two things that make us so that we don't have as much water gain and that we have an increase in water loss. And so that mechanism of ADH is shown in that previous slide, slide number 5. What we also have, though, is that what this does is that then we end up peeing off water. We keep the salt, meaning we keep the sodium in the nephron, but we lose water through this mechanism that's outlined here, where we basically put in water pores and insert water pores into this the lumen side of the collecting duct, and then what that does is that ultimately allows this water to be able to be absorbed. And so this is the case with antidiuretic hormone being present. However, what can happen is that when you have a drop in ADH, you lose these water pores that are present here on the lumen side here. Fewer water pores mean less water gets absorbed back into the blood from the nephron. So that's kind of the gist of it. So what's happening then is that you're losing more water. It's staying in the lumen of the nephron. It's going straight out through the and getting peed off, basically, until you finally restore your homeostasis. So that's how a response to osmolarity. But there's another kind of thing that can happen, a different type of reflex, and that one's tracking blood volume. And in this case, what happens is that we are talking about where we have homeostasis, where we have a normal amount of extracellular fluid. And the thing to try to remember is that in order to get rid of that blood volume, you've got to get rid of the sodium and you've got to get rid of water. So in the previous case for osmolarity, we just wanted to pee off the excess water that we had already had. But in this case, what we're doing is that we're trying to actually get rid of both the water and the sodium, because we need to get rid of both in order to decrease our blood volume. If we only got rid of water here, it would make us really, really salty. So let's go through this type of thing here. So what we're talking about here, we are at homeostasis, and then we've increased our extracellular fluid and salt gain, which then causes an increase in blood volume and distension of the atrium. The atria releases what's known as AMP, or atrial natriuretic peptide, and that in turn then coordinates two different effects. One is that it drops ADH. We talked about what happens with ADH. You pee off water. The other thing is that it decreases aldosterone, and aldosterone, as you guys are learning in 290 probably about now or just or a while back, there was that aldosterone is important for helping to control and keeping sodium retention. So you're losing your water retention hormone here, you're losing your sodium retention hormone here, and so those two factors are making it so you lose your water, you lose your sodium, and in this case, you're peeing it out. It stays in the lumen of the nephron and then ultimately ends up in your bladder and out, out into the toilet. And then and then homeostasis is restored. So a couple of things to keep in mind as you're navigating this renal stuff here is thinking about what the hormones do, like aldosterone. Think sodium retention. That's the easiest way to remember it. ADH, or antidiuretic hormone, is all about water retention here. If you can remember it those two ways, it really helps to be able to do it. AMP, or atrial natriuretic peptide, just think about what happens like in the Bainbridge reflex, where we have a stretch of the atrium and that causes a reflex firing. In this case, we're talking about how the atrium releases this AMP when it has a lot of blood volume, and that helps to coordinate this effect. So those are the two pathways that are in play here. So how does this relate to our experiment? Because this is a question that can come up. In previous cases, it's come up, and it's important for us to be able to understand how this works. So if we look at what happens in the case of the effect of osmolarity, we're comparing three different types of conditions here. One is we're looking at, we have one group here that drank 1 liter of pure water. And this is the volume, and then this is over time. So if they drank pure water, basically, you can see that they had an increase in the amount of volume of urinary output at about an hour. If you had them drink 1/3 of an isotonic solution, 1/3 of a liter of isotonic saline solution, you see that there's no real change in what they've peed off. And then finally, the control condition, where they didn't drink anything, is running a little bit lower. So we call this the effect of osmolarity, but I think the thing that's a central question that people get really confused about is, how does comparing 1/3 of an isotonic solution here to 1 liter of water up here, how does that comparison have any meaning at all? And the short answer, which I'll just tell you verbally before we kind of go into the details, is that when you drink pure water, about 1/3 of it stays on your extracellular fluid to extend and increase your blood volume. When you drink 1/3 of a liter of the salty isotonic solution, what happens is that you end up only increasing your extracellular fluid, or your blood volume, by 1/3 of a liter. So what's the difference then? They both increased your blood volume by 1/3 of a liter. Well, in this case, you have salt along with your water, so there's no change in osmolarity. Whereas here, you've increased your blood volume, yes, and you've also dropped the osmolarity. Two different factors. And so what we're looking at is the interplay of two different reflexes here. First one here for osmolarity, second one here in terms of blood volume. And so the question becomes, well, if we increase the blood volume in both cases by 1/3 of a liter, then, here we're going to see this kind of an effect here. And maybe, that's the dominant type of reflex that's important for making us pee when we drink 1 liter of water. But what we might find out, though, is that really this osmolarity reflex, the sensitivity to osmolarity, is really the key thing to help drive us in driving us to increase our water loss and urinary output. So the diagram that I've created for you guys is over here, and this is also on Blackboard for you. So what I did is I drew a schematic illustrating the intracellular fluid and extracellular fluid. And then I illustrated several different types of changes here. We're going to compare this one down here and this one over here. So 2/3 of volume of your blood volume or of your fluid is in the ICF, 1/3 in the extracellular fluid. 0.3 osmoles on both sides of it. So if you drink 1 liter of water, then what happens? What happens is that 1 liter of water, 2/3 of it goes into the ICF, and 1/3 of it stays in the ECF. And this is really weird because people think about this and they're like, well, why would it split up between those two different compartments? And the way to remember it is that basically this 1 liter of water is diffusing across the cell membrane, and it doesn't see the cell membrane. The cell membrane is permeable to water. And so it just diffuses evenly out between these two compartments. And this one's 2/3 as big as this one. What it's also going to do, though, is in addition to expanding those two compartments by their proportions, it's also going to dilute the concentration of the solutes in solution. So the osmolarity is going to go down. So I drew here this little sign. It's just like what your body sees. So it's like a little eyeball basically trying to say, and it says, okay, look, blood volume has increased by 1/3 of a liter. So it sees that and it's going to activate a reflex to do it like we talked about on the other slide. This shows the other eyeball, another reflex that's basically tracking osmolarity, and there's a drop in osmolarity. So what happened here? Well, here you can see that there's like 325 milliliters of output that happened here. What happened? Osmoreceptors dropped their firing. That caused a decrease in ADH and a decrease in water retention. Now the blood volume reflex was also in play here, and so and so how do we discriminate that? How that and that kind of impact, because right now I'm just describing everything that happened with osmolarity, but what about this factor here? Well, what we can do is we can say, well, let's take a condition where we only increase the blood volume here and didn't touch osmolarity. And so when you drank 1/3 of a liter of isotonic saline solution, then what you found was that you increase your blood volume by 1/3. And so you see this expansion of the extracellular fluid in this case. No change in the volume or of the intracellular fluid, and no change in the osmolarity. So that's shown here. There's a little, that's supposed to be no change sign right there. And so then what that means is, and what we saw is that there wasn't very much output there. And so even though we had an increase in blood volume by 1/3, the activated stretch receptors caused AMP to decrease sodium retention and water retention to make us pee there. What you can see is that there wasn't as dramatic of an impact as what we saw over here. So when we compare these different conditions, the unique factor here is the osmolarity, and that the osmolarity dropped. So be prepared to be able to describe that scenario and what it means in the data that you've collected. Another scenario that you guys tested was that you looked to see what would happen when you compared 1 liter of isotonic saline solution to 1 liter of Mountain Dew. And so kudos to those of you who drank Mountain Dew. Dr. Pearl said that you guys were a little bit reticent. I'm glad you guys did it, though. It's well worth it to do the Dew. And what you can see is that with caffeine, typically what you see is a huge increase in the amount of output that happens here. And so there's a couple of mechanisms that are in play, and the one that we're trying to illustrate here is that there's a drop in ADH. So back in the day, before I was here, they used to actually give alcohol because the drinking age was 18. And so they would give it to the students who were above 18 and give them a beer. And this is a more pure effect of alcohol in terms of causing a drop in the antidiuretic hormone. But now we have to substitute it with Mountain Dew, which does a couple of things, including increasing blood pressure and some other factors, but can have this kind of effect where we see this really big increase in it. So if you drop your ADH, then what you're looking at is you're looking at how these different factors of aldosterone start to get affected. So in this case, if you're artificially dropping antidiuretic hormone, then you're going to end up losing more fluid. You're not going to retain as much water. So that's kind of what's in play here, and what you're seeing when you see this. Well, you compare this to 1 liter of isotonic saline solution, and the principle was basically that Mountain Dew and the 1 liter of isotonic saline solution, that they're both isotonic. I think we've actually, recent tests have shown that this might not be as isotonic, but let's take a look then and see what happens if we assume that they're both isotonic. Then you drink 1 liter of isotonic saline solution, and again, kudos to those of you who did this. And what you find is that there's an expansion in the extracellular fluid of 1 liter. So both of these conditions are going to make you pee more. Which one's going to have the biggest effect? The caffeine, or where you increase your caffeine and blood volume, or the case where you just increase your blood volume? And so in this case, what we saw is like, about 250 mLs of output was seen when we had an increase in blood volume per 1 liter. Whereas if we have the Mountain Dew, we add the caffeine into the mix, we increase it almost twofold. So that's the kind of effect that we're seeing with this, with caffeine. And so, and that illustrates some of the different types of factors that we see here, right? So Mountain Dew is being shown here, where there's an increase and it keeps going and stays up for a long time at this case. And then here is where we had the isotonic 1 liter of saline solution, which is making you pee more, but isn't quite as dramatic as what you see here. So these are samples from last year's lab, or maybe it was 2 years back, but just an example of some of the types of output that we had. And then I have there on Blackboard for you to be able to just take a look at and get. So, so go through that. Go through in the lab notebook, make sure that you're going through each of the different points that they have at the beginning to make sure that you understand what the types of questions are in play for the final that's coming up. So you're ready for that. All right, thanks. And I have office hours on Tuesday, and I'll be in Henry Hall there to be able to help out with asking any questions. And then we'll be meeting up at 3:30, and I can take questions at the very beginning for the first 10 minutes or so before we actually begin the final. And again, just to remind you, the final is only covering everything after skeletal muscle. So it's heart lab, the frog heart lab, onward. That's up for grabs. So go through those reports, make sure you can do everything. Go through the lab notebook and see how they actually have all of those graphs of how to actually be able to do your measurements and the types of questions that are in play. And the last point that I'll say for the final is that when you're doing the blood pressure, that one where we did the report on blood pressure and we did all those measurements of stroke volume and of cardiac output, mean arterial pressure, those types of things. Go through that. We have on Blackboard some summary of those different things. I'll show you there. It's important that you know those equations. And because you did a report, sometimes in previous times they found that makes it a little bit, people are a little less, they don't remember it quite as well. So let me point you to that. That is right here, blood pressure, heart rate, respiration. And then you can go through some of the notes that we have here in terms of talking about the Valsalva maneuver and some of these changes that happen with those different equations. And then another useful one is here, where we talk about it. It's much more cleanly written and talks about these different equations here. So master these equations and these points here to make sure that you really nail the key things that you have for the final. All right, I'll close with that. Thank you so much. And again, let me know if you have any questions.